Okay, so this sheet is part of your notes. This is going to walk you through how to work a Punnett square problem, or a genetics problem using Punnett squares. So you've got a problem at the top, and it says in pea plants, red flowers are dominant over white flowers. You've got a heterozygous red flower, and it's allowed to self-pollinate. So that just means that pollen from the plant is going to move on itself, okay, to the egg cell. So pollen's going to contain sperm, and then that sperm and the pollen grain is going to move to the egg on the same plant. Then it just asks you for the genotypic and phenotypic ratios that are possible, okay, or that are going to be the outcome in crossing those two parents. So these are the steps that you've got to follow. This is what I need to see when you're working genetics problems. So first you're going to choose a letter to represent the genes in the cross. You've got to have a capital letter, of course, for the dominant alleles and a lowercase letter for the recessive alleles. Make sure, you've got 26 choices as far as letters of the alphabet. Make sure you're choosing letters such as R's, T's, B's, things like that. An S is not great unless you're going to make a giant S and a tiny S. Okay, a C is not great unless you're going to make a giant C and a tiny C. And then for step 2, you're going to write the genotypes of the parents, okay, with a little X in the middle. And so that means that it's crossed with. So since this is just self-pollinating, in this particular example, both parents, quote unquote, even though it's the same plant, are going to be heterozygous with one dominant and one recessive allele. Then your third step is where you are going to show these diagonal lines that you can see there. This is showing the separation of the homologous pairs during meiosis. Okay, now eventually the little lines I'm going to say I don't necessarily need to see. All right, so this is kind of a step that gets you to step 3, gets you to step 4. Right, so we are showing the separation of homologous pairs during meiosis, specifically that those homologous pairs are going to separate during anaphase 1 of meiosis, so that each new cell by the end of meiosis 1 has 1/2 of each homologous pair, okay? Or we can talk about in all reality that we're separating, never mind, I'm not even going to say that, okay? So then you're going to take these alleles that are possible in the gametes and write them for each parent, okay? Let's call this the parent that's on the left here, and this the parent that's on the right there, okay? Typically, you're going to write the male, if you know male versus female, male alleles are going to go on the top and female on the side, which I think we may have already talked about in the video lecture. Okay, if you don't know, then it doesn't really matter. All right, but that's just kind of standard convention. Okay, so then you're going to fill in the Punnett square, fill in the boxes. So again, these are alleles that can be in either egg cell over here or sperm cell here. Okay, remember there's going to be 4 sperm that are produced, so 2 of those 4 would have a capital R for a dominant allele, and 2 would have a lowercase r for a recessive allele. Okay, so if we can just imagine that, 1, 2, 3, 4. Oh, sorry, that last one is messy. Okay, and then over here for egg cells, 1's going to be big, 3 of them are going to be small and bipolar bodies. Okay, so this one could be obviously either capital R or a lowercase r, same kind of thing. Okay, so if it's a capital R, when we combine a sperm and an egg with a capital R, then we get a homozygous dominant genotype in the offspring. So again, outside parent alleles, inside possible allele combinations in the offspring. Okay, so here we're going to have homozygous dominant genotype, a heterozygous genotype here, another heterozygous genotype here, and homozygous recessive genotype here. So for example, that little r and that little r make that box. That little r and that big R make that box. Okay, so then step 6 says that you're going to determine the phenotypes present in the offspring. And so all of the possible combinations happen to be in that particular Punnett square. So those are written out with the phenotypes that they might code for, either being red or white. Okay, and then those are paired up. Okay, that genotype homozygous dominant codes for red, so does the heterozygous genotype, and then the homozygous recessive genotype. Then following that, you're going to write your ratios, okay, your genotypic ratio which I very often abbreviate to just GR and PR when we're working problems, okay? So your genotypic ratio and the alleles that, or sorry, the genotypes that go with that are split up here. Same thing with the phenotypes, right? When I write ratios, I combine them, okay? If you need to split them up, that's fine. If you write just numbers though, okay, let me just cover that up. Oops, pen rolled over there, okay? If I just have 1 to 2 to 1 or 3 to 1, those ratios are meaningless without the genotypes or phenotypes with them respectively, okay? So, then if you just did this and just wrote those are meaningless as well. So it's going to automatically be wrong if you do not give me the combination, okay? So your genotypic ratio, if you combine them, would be 1 to 2 to 1 written like this, okay? Then your phenotypic ratio would be 3 red to 1 white, where those 3 red are the homozygous dominant and the heterozygous genotypes. Then there's your 1 homozygous recessive genotype, okay? And every time you cross 2 heterozygous individuals, you're going to get this particular outcome. It's never going to change if they're heterozygous and we're following Mendel's rules, okay? Then on the bottom of this, you've got 2 practice problems, okay? And just kind of depending on the situation, that I may or may not tell you to do these at a certain time, as when I'm doing this particular video. I don't have a plan for that yet at this time, just recording how to walk through the steps of a problem. All of your work has to be shown, okay? When I tell you to complete a Punnett square problem, I need to see steps 1 through 7. Like I said, eventually I'm going to pull step 3 kind of out of there, all right? But you've got to understand, if I'm giving you a problem on a test or a quiz, that if I were to tell you to write the cross, for example, of the parents, that's what I mean to do, okay? So if I were to say, okay, for part A, you have to write the alleles and what phenotype they code for, okay? Then if I said write the cross between the 2 parents for part B, if I then said, because eventually I'm going to pull this part out of there, let's say I skip to complete the Punnett square, all right? Then yes, you would start here, but this would be what I would see as my final product there, hopefully, right? And then following with step 6 and 7. Okay, if you have any questions about how to work problems, let me know.